Yes, we're very excited to present these results today. If you guys have the interim report, if anybody needs that, we'll send copies. Some of this was in there, but we do have additional results since then that we're very excited about. And fortunately, a lot of them support our hypotheses that we were previously working on. So some of this stuff was covered pretty extensively in the report, but just really quickly, here's the results of the citizen science campaign in 2019, and you can just see all these blue dots on the map are where sightings were reported, and these red dots are where we actually went out and took foam samples. So we got a lot of reports coming in September, October, and November, and then they started to decrease a little bit in frequency. Our main question was really, as Sally mentioned, what is the source of this foam? That was really the first thing that we tried to focus and concentrate on. So we started off by thinking about what potential sources to the foam could be. Looking in the literature, foam is not always created by the same thing. So you can't just assume it's one thing. So we started off looking at streams, anything coming in from the terrestrial environment, watershed. So we looked at soils, we looked at for agricultural signatures, and of course we looked closely at the invasive mussels, in particular the quagga mussels, which every time we went out to collect mussels, we found those were dominant. We had problems finding a lot of zebra mussels. We looked at aquatic plants, macroalgae, seaweeds as they're commonly known as, so things like eelgrass, and we also looked at the free-floating macroalgae species. So like every time we went out and collected foam, there was always floating duckweed and watermeal in it. Really to look at those too. And finally, we looked at phytoplankton. And in particular, we were focused on Microsystem, so the harmful cyanobacteria that Canandaigua Lake is having some problems with. You can see on the map over here, the yellow pins are where we collected foam. And so some of these were shoreline samples, some were mid-lake samples. So Lasher LA 927 was the one that Stella and I and Lindsay went out with Marty Lasher to collect. And we tried to get some at the southern end, we tried to get some at the northern end, and we got pretty good coverage. These blue pins are where we collected stream samples. So Stella and I drove around the lake and we collected a bunch of stream samples. And we also collected biological tissues. We did a plankton toe. We went out and collected quagga mussels, and then we collected these macroalgae samples as well. Very briefly, how we treated the foam. It was a real process figuring out how to sample and how to deal with the foam in the lab. But what we ended up doing is collecting these buckets, letting the foam sample into a water sample. We collected a nearby lake water sample to compare with the foam. And everything was filtered through a 0.7 micron filter. And that's just in the field, that's how we determine what is dissolved in the water versus what is particulate stuff. You put it through this small filter, everything that goes through is dissolved, everything that doesn't is particulate. So we're really focusing now on the dissolved data. So the dissolved chemistry is what is allowing foam to be created when the water gets aerated. So you got to have the right chemistry at just the surface of the water, and then you get wind coming through and stuff, and it starts to create foam. What we're looking at here is just the carbon and nitrogen data. So the simplest look that we can take looking at this. What we see on the y-axis is the concentration of nitrogen in the water sample. On the x-axis, it's the concentration of carbon. These are the lake water samples. As you notice, they cluster together very well. They're low in nitrogen, they're low in carbon. It's definitely what we would expect from an oligotrophic / mesotrophic lake. The stream samples, moving up along the y-axis, are elevated in nitrogen relative to the lake water, but they have similar amounts of carbon. The foam samples, it was the opposite. The foam samples have similar amounts of nitrogen as the lake water, but they're very enriched in carbon. I didn't circle this sample by Lynn Klotz. That one was allowed to sit in the lab for a period of time, so we could see the effect of sample storage and degradation. So it's useful for us, but it's not treated in the same way as the other samples. So we see a strong enrichment of carbon in the foam. What you're looking at here is this is how we typically look at sources. We make a source plot. So we took that carbon and nitrogen data, and we took the ratio of carbon and nitrogen and plotted it along the y-axis. Carbon and nitrogen ratios, as some of you may know and have seen and looked at, they're really useful for looking at sources. Phytoplankton have really ratios like 7. Land plants can have much higher ratios like 20 or 40 or even higher. And we plotted those carbon and nitrogen ratios against carbon isotopes. So this, what you see down here with this delta notation and the 13C, what we're looking at is the weight of carbon. So most carbon weighs 12 on a periodic table, but a very small percentage weighs 13. And different types of plants and primary producers create different signatures of these carbon weights. So to keep it as simple as possible, what we see where most of our samples plotted, most of the end members we collected in the foam, all very depleted, very light carbon, around -27 to -31. We think that most of this is being fed by the bedrock in the area, which probably contributes this light carbon, and then it's just recycled through the system. With the isotopes, a couple samples where I want to point out is covered by the Zoom video, but over here, you see that C4 plants like grasses, corn, or C4 grasses like eelgrass, stuff like that, they have very different carbon signatures. So we can rule those things out with the carbon signatures. Everything else looks kind of the same. But what was really interesting was that the carbon and nitrogen ratios of the foam is incredibly high. I mean, much higher than we expected. So our source plot didn't work the way we would expect it to because the foam looked so different. And so what we think we're seeing is that because the foam isn't a couple of these sources mixed together, the foam is the enrichment of a particular chemical from one of these sources. So that's what we're now seeking to identify. What could this possibly be? Why does it look so different? Just as a side note, that's all the dissolved data, which we're going to be focusing on. We also kept our filters. We weighed them to get total suspended solids, and those are sent out to a lab that is closed. They're hopefully reopening in the next couple of weeks where we can get that data, but it's less important than the dissolved data because the particulates are not causing the foam. They just get caught in the foam as it moves. But what we see is the total suspended solids in the foam were 100 times those of nearby lake water and 20 times those of streams feeding into the lake. So the foam captures a lot of stuff once it forms. That's kind of the take-home message there. Floating plants, dust, algae cells, things just get caught up in it. Okay, so looking at the carbon and nitrogen, we were able to start ruling out some things. Humic substances in soils. If you were to, put a bunch of water on some soils and see the water coming out, it looks like tea. That's like what humic substances in soils. The C to N ratios don't support those being a source. And that's important because when we look in the literature, a lot of natural form is formed from humic substances. So we definitely wanted to rule those out. We can rule out C4 agricultural inputs because the C13 ratios were so depleted. We can't necessarily rule out C3 inputs, but overall the terrestrial contribution, as we'll continue to see, looks very small. And we were able to rule out any submerged aquatic plants that are grass-like, things that have C4 signatures like the eelgrass. So this leaves us with kind of what we were focusing on anyway, which was the invasive mussels, the macroalgae, now particularly these free-floating species, the duckweed and the watermeal that we found in a lot of these foam samples, and of course phytoplankton. Okay, so when we're looking at the carbon and nitrogen ratio and we're looking at the carbon isotopes, we're looking at all the organic compounds in a sample. What we do now is what's called biomarker analysis. When we say we're going to actually zoom in on one particular type of compound, and we're going to see what its distribution looks like. So we chose lipids and fatty acids, and we did that because they are surfactants and they can create foam, so they were the natural thing to look at. Plus, they're commonly used in aquatic systems to differentiate sources. What we found is that the lipid yields from the foam were very low. We didn't expect that. What that told us is that the lipids are not causing the foam, although they can still point us to the source. They still contain a lot of source information. One of the interesting things was that all the fatty acids that we found in the foam were in the form of triglycerides, which is how fatty acids are stored as energy in cells. And this just means that our sample was very fresh. It contained very fresh biological material. So in the lab, we did a process called saponification, which just breaks apart these triglycerides. And we looked at the distribution of fatty acids that were released. And they contain very important information on the sources. Here is a chromatogram from the instrument we used, which is GCMS, or gas chromatography mass spectrometry. But the point is that fatty acids differ in their chain length, and they differ in the amount of double bonds. So if you look over here, this compound on top is shorter chain than the compound on bottom. That contains important information. Moreover, is there an even amount of carbons or is there an odd amount of carbons? That's important information. And also, how many double bonds do you have? So this fatty acid on top has no double bonds. That's what we call saturated. And that's important because we'll be mentioning saturated fatty acids again. This compound on bottom, oleic acid, has one double bond, so it's unsaturated. So that's all that saturated and unsaturated refers to. These were the fatty acid distributions we got in the foam and in the biological tissues we looked at. So all these highlighted yellow boxes here, this is the foam data. We took an average of all the foam samples, so it was easier to look at. So what you're looking at is each one of these bins on the x-axis is a different type of fatty acid. The ones over here on the left are really short chain fatty acids. The ones over here on the right are longer chain fatty acids. And what we can see is that the foam contained mostly short chain, mostly saturated fatty acids, and they were mostly even chained. They did not, C16 is the most dominant fatty acid, but that's nonspecific. We found that in everything we measured. But there were other types of fatty acids that had higher relative concentrations in the foam compared to any of the biological tissues we looked at. So the point here is that the fatty acids in the foam did not look exactly like any of the tissues that we measured, but they are extremely consistent with reports from Microsystem biomass in the literature. Microsystem primarily make C14 to C18, only even carbon chains, and mostly saturated fatty acids, with the exception of some C18 unsaturated fatty acids, which you see right here and what you see right here. So this is all very consistent with what we found. Now our plankton toe was a mix. There was Microsystem in there, but there was also other types of phytoplankton. There was zooplankton, whatever it picked up. So that's why this can look like Microsystem, but not look like our plankton toe. Now we're just gonna, so what we can do is, it's one thing to look at this profile, but we want to distill this down into some numbers that will tell us something about where these fatty acids came from. So the one thing we're looking at is the terrestrial to aquatic ratio, right? We were looking at stream sources, agricultural, terrestrial sources versus aquatic sources like phytoplankton, aquatic plants. So what this ratio does is it looks at aquatic fatty acids, which are typically short chain. So it uses C12, C14, and C16. And as we can see in our foam, plus the other types of biological tissues we looked at, these were the dominant fatty acid chains. The terrestrial chains, which is C24, 26, and 28, we didn't find any in the foam. We found none in the macroalgae and none in the plankton, which makes sense because they're aquatic sources. We did find some in the mussels, but in low amount. But the point here is for the foam, we get a terrestrial to aquatic ratio of 0, which means that we can now say that the fatty acids in the foam represent a very fresh aquatic source.
Thank you.
There's one more thing we can look at that gives us a lot of information, and that is the odd chain. So everything I was talking about before, these terrestrial and the aquatic, they're all even chained. These odd chained fatty acids tell us how fresh the sample is and if there's any microbial inputs. And we can also calculate something called the carbon preference index. What this shows is this number becomes really high as you have mostly even chain fatty acids, and it becomes lower if you have more odd chained. And you'll see, so up top here, these are all values from the literature. In the bottom here, these are all values that we measured in the study. And you'll see our foam were the highest. They were much higher than any of these other published sources. They were the closest to the phytoplankton. And actually, if you look at Microsystem here, their value is indeterminate because they only create even-chained fatty acids. They don't make any odd ones. And that's why we think our foam value is so high. And in fact, this is an average. Some of our foam samples were also indeterminate. So once again, the fatty acids seem to point to a fresh aquatic source, in particular, Microsystem. It's consistent with Microsystem's biomass. And the lack of odd chain just show that we didn't get any storage artifacts. These samples are very fresh. While we let the foam collapse, we don't believe we saw any artifacts, and there's minimal inputs from bacteria. So we can take the initial sources that we thought and distill it down farther. The fatty acids, they just didn't show any terrestrial material in there. So we can rule out stuff coming in from the watershed. They also make the invasive mussels and the macroalgae less likely because we did not see fatty acids that were as consistent with those sources. But we really want to be sure. And this is where we left off at the interim report. And now I'm about to show you the new data that we have that helps to support some of these hypotheses. Okay, so this is FTIR, or Fourier Transform Infrared Spectroscopy. What this does is you actually create an image of your sample. So you take your powdered sample or your water sample and you shine infrared light on it. And some of that light gets absorbed and some of it passes through. And then you map that out for different wavelengths. And the reason we do this is because certain classes of molecules absorb different wavelengths. What I've got plotted here, this yellow line is a fresh foam sample. This was one of Marty Lasher's samples, and it was collected as foam was forming. This green line here is one from Chuck Wichelli, and that one was taken from the shoreline, so it represents an older foam sample, possibly a more degraded foam sample. What you can see here are characteristic bands. So this is all the lipids in the sample absorbing the infrared light. These peaks get bigger if there's more lipids, and these peaks look relatively small. Over here, we can see where proteins are absorbing light. And what we see is that the fresh Lasher sample has very small protein peaks, and the older Wichelli sample has bigger protein peaks. So what we think that shows is that proteins are accumulating in the foam as it ages, but they are not an inherent part, a major inherent part of the foam when it first forms. The real interesting part of the story, and this is what we think those weird C to N ratios were telling us all the time. We've got to have a concentration of a particular chemical component that's really enriched in carbon. It has very low nitrogen, and we think that's carbohydrates. And that's exactly what this FTIR shows. Both of these foam samples, the FTIR spectra, are dominated by the carbohydrate peaks. So let's look at the Lasher. Once again, notice that this carbohydrate region is the biggest peak in the chromatogram. The proteins are relatively small, and the lipids are also relatively small. So let's compare it to the biological tissues that we looked at. First, let's take a look at the Quagga mussels. Here's an image from the Quagga mussels. Notice their protein peaks are very large, much larger than in the foam. And their carbohydrate peaks are relatively smaller. So in the mussels, we see the exact opposite of the foam. The foam is very enriched in carbohydrates, not so much in proteins. The mussels are very enriched in proteins, not so much in carbohydrates. Let's take a look at the plankton toe. Now, the plankton toe is strong in both of these regions. You've got big protein peaks, and you've also got a very big carbohydrate peak. But that makes total sense. When you're looking at bulk phytoplankton, they make proteins, and they make carbohydrates. But they have a lot more carbohydrates than the mussels to potentially give up to the system. When we look at the watermeal and duckweed, the eelgrass I'm not putting on here because the eelgrass we already ruled out. When we look at the watermeal duckweed, the protein peaks are very small and the carbohydrate peaks are very big. So this is a potential contributor to what we're seeing. But that's why we need to zoom in on the carbohydrates only. So now we've shown that just like we thought, the foam is primarily carbohydrates that's forming it. And multiple of these sources have carbohydrates to give up. The mussels, not so much. But let's look and see if we can look at this carbohydrate profile. So what we did is we took it and we zoomed in on it. Each one of these boxes has this yellow peak, which is from that Lasher fresh foam sample. And I've overlaid different things on top of it. Each of these blue lines here represents an important part of the spectra to look at. These are where different polysaccharides, which are the type of carbohydrates we're looking at, this is where they would give peaks in our spectra. And what we can see is right here, even though Shelley had added more proteins because it was aged, the carbohydrates in both foam samples looked very similar, which again is consistent with the carbohydrates forming the foam because that's why they look the same. They're not randomly accumulating these based on where they are. The mussels look similar in certain areas, and in other areas of the spectrum, they diverge from the foam. The macroalgae, which had a big carbohydrate peak, the carbohydrate peak looks very different than the foam carbohydrate peak. And what we see over here finally is our plankton toe on top of the Lasher sample, and this gives the most agreement in terms of the carbohydrate peak. Now there are some small differences, but once again, we think that these carbohydrates are coming from Microsystem in particular, and our plankton toe was a mixed community. So let's take a step back again. All these organic compounds, right? So it's not just what organism is making the foam, but what's in that organism that's making the foam. Can we pinpoint it? So these are all the different types of organic compounds that you would normally look at. We know it's not these two. They're not formed in high enough abundance. They break down in the environment. So carbohydrates, lipids, and proteins. Well, we've now ruled out lipids. We got very low yields when we extracted them, and the FTIR showed very low concentrations of lipids. Proteins have a lot of nitrogen in them. That's not consistent with the carbon-nitrogen values, and also the FTIR shows that the foam doesn't start off high in protein. It gets it as it ages, so the protein's not forming it. All our data point to carbohydrates, in particular polysaccharides. Polysaccharides are long chains of sugars that are formed by organisms for different purposes. They're known to be a big component of the DOC pool in aquatic systems. And we think that the Microsystem algae is probably exacerbating this. So, and I'll get into that in a second, but let's look at take one last look at our source chart. We can rule out the mussels. It's very unlikely they're the dominant component in there. It's supported by fatty acids. It's supported by the FTIR. The macroalgae, now we think that the macroalgae still could be contributing some carbohydrate, but it does not look like the carbohydrates we're seeing in the foam. So they're probably not the dominant component. And that leaves phytoplankton. In particular, our data is consistent with what you would expect from a Microsystem source for the biomass. The fatty acids showed a fresh aquatic source. They were very consistent with reports of Microsystem in the literature. The FTIR and the C to N showed that we have high concentrations of polysaccharides in the foam specifically, and that those polysaccharides look like phytoplankton. And also anecdotally, from everything we've heard, the foam formation coincides seasonally and historically with these harmful algae blooms. So what does that mean? What is polysaccharide from Microsystem algae? What does that mean? Why is it there? Well, when we started to look into this, it all came together and made a lot of sense. Microcystis forms these very thick concentrations of what are called exopolysaccharides. So they're polysaccharides, they're sugars linked together, and the microcystis makes them and it releases them into the environment. That's why they're called exopolysaccharides. And these pictures from USGS, for those of you who have seen microcystis under a microscope, you'll see them in these colonies. And these colonies are glued together by these exopolysaccharides. So they make these to form colonies. They make high concentrations of it. It's also known as slime in the literature. It's very slimy. And they use it to regulate their surrounding environment. Studies have shown that during microcystis blooms, about half of these polysaccharides that are made end up entering the DOC pool of the lake. So they're forming this stuff, and then they're releasing it. We think we actually identified and saw this compound in the lake. When we were doing fatty acid extractions, we kept forming these emulsions that were very difficult to deal with. When we started looking in the literature, these emulsions are characteristic of exopolysaccharides, and they're actually used in things like the food industry to create emulsion. So we think that's exactly what we're seeing here. Interestingly, we also formed an emulsion, even though the quagga mussels weren't consistent with our source metrics, we did form an emulsion here when we extracted the quagga mussels. And so we started to think more about the role that quagga mussels play in this whole thing. So the microcystis algae are not existing in their own world. They're a product of the entire ecosystem. Now, the quagga mussels, as we already know, are responsible for the, over time, increasing concentrations of microcystis in the lake. The mussels eat other types of algae. The mussels are constantly filtering the lake, and they're eating the diatoms, they're eating other types of algae, and they spit the microcystis back out. So when you think about the time scales that they filter the whole lake, you selectively get microcystis over time. The interesting thing is that quagga mussels actually get sick when they eat toxic algae, and they start to form more mucus. So what you can see on top is a normal, healthy quagga mussel feeding on diatoms. And on the bottom, you can see a quagga mussel feeding on toxic microcystis, and you have extra mucus releases that are very atypical. So what we think happened is that the mussels are not contributing the bulk of the biomass to the foam, but they are playing an important ecosystem role in having this foam formed by selecting for the algae that makes these polysaccharides and by recycling these polysaccharides. So they may feed on these microcystis and expel these polysaccharides back out as opposed to them breaking down, and that's why they linger in the system. Okay, so that is the source story. The final thing we looked at, which may have a little bit to do with source, but it's more about the toxicity concerns of the foam, was the microsystem analysis. We found that the foam typically had higher concentrations of microsystem than the surrounding water. Typically unfiltered samples, so total microsystem in cells and dissolved in the water, were higher than for the filtered samples. And really, so here we showed the drinking water limits, we showed the recreational water use limits, and we can see most of the filtered lake samples were below. And it's important to know that not at one point we were never sampling in a bloom. Like, we weren't really expecting super high numbers. We weren't sampling lake water that was in a bloom. This was just normal, clear, late summer looking, lake water. And we got many hits in the foam above the water. But the most interesting thing is that I think that there's more work here to be done, because this was one of Sally's samples, shoreline samples that she took for us. And this one had values of microcystin that were above the recreational water use limit, even though you can see down here the amount in the nearby lake water was very low. So you wouldn't have detected it in the lake water, yet the foam was above the recreational water use limit. And finally, this is the sample that Stella and I took with Marty Lasher and Lindsay. This mid-lake filtered water sample gave the highest concentration of microcystin, about 38 micrograms per liter, even though down here, the filtered lake water was under detection limits. So I don't think the full story is out yet on the microcystin concentration in foam. What this shows is that there's often higher concentrations in the foam, but there are some situations where you can have definitely a high enrichment. And I think it would be interesting to know what the foam looks like if, say, it was taken near a bloom, which none of these were taken right next to a large identified bloom with very high microcystin concentrations. Okay, so to sum everything up, the main conclusions of this work is that the foam chemistry was similar among events. Different foams collected look very much the same in a lot of ways, but they tended to change as they aged by picking up more material. Microcystis algae is the most likely source of the foam. We have several separate pieces of evidence to support that, and we think the mussels are not contributing the material directly, but they're playing a very important role in this. And I think there's much more that can be studied on that front. And foam has the potential to concentrate microcystin. We don't know always to what degree. We would definitely have to have more data on that, but it does show that it tends to be higher than the surrounding lake water. So as Sally pointed out at the beginning of the talk, she's absolutely right. The more, and that's just the nature of these science in general and these types of studies, the more questions we answer, the more questions we're going to have. The obvious next steps would be, let's isolate and characterize this polysaccharide. What is the nature of this polysaccharide? Do microcystis always make this type? Is it related to their environment and things such as that? Have we missed anything? Is there, say, a terrestrial input that we have missed in the streams? Have we missed some sort of a pollutant input? And I bring this up because this is a really critical point that I'd like to make. What we've done is we have characterized what's in the foam, and the foam looks like it is mostly carbohydrates from Microsystis algae. But that falls short of showing a direct link. There is a small chance that what's going on is that we're seeing all these carbohydrates from the Microsystis algae, and it's priming the water to be foamed in a way, but it's not foaming yet. And then comes into the system some small amount of pollutant or something else that is present in smaller amounts, but it changes the chemistry fundamentally so that foaming can occur. And I'd say the only way you could answer that is to just by broaden our look for the types of things we're looking at. I know PFASs is one thing we have mentioned, and we have a contact with the lab who does that, so that might be something to look at in the future, as well as organic phosphates that are applied to agricultural fields. It would be really interesting to look at those in more detail as well. More microcystin monitoring. What's more of this foam concentration of microcystin story? And of course, eventually you would take all this exploratory research and you would move it to the lab where you could recreate these conditions and see the foam being formed from a microcystis culture that has the same signature. So this would be the natural progression of this study in the scientific field. So steps moving forward, which is kind of the question probably in everybody's mind. Obviously, the completion of the project has been a little bit by lab closures. Fortunately, we've been working with Brockport to reopen, to get the lab there back going. One of the labs we're working with will hopefully reopen in the next couple weeks for the particulate data, but we feel the particulate data is less important. So what we would like to do is get the PCB data, which is the final toxic component of this study, get the PCB data when Brockport opens, and write and submit the final report, and then work with you guys in terms of how you want to disseminate the results. Is there anything we can do to help in terms of like outreach or creating fact sheets or anything like that that you guys would want help with? And then it's, what's the next thing? Is there funding through CLWA to move on to the next step of this research, or just do more foam monitoring or more collection. We're also looking at proposal opportunities with the caveat that if we start writing proposals now, we think we have a very important seed data. This is a really riveting story in a lot of ways, and I think it'll have broad scientific interest. But proposals are slow, multiple submissions. It could be a couple years before we're funded on a proposal for this work. We would also like to start reaching out to other watershed associations, and maybe that's something that you all can help us out with in a way, because as we start to expand beyond Canandaigua Lake, we can start putting it into context and getting more pieces of this puzzle solved. So I'm going to leave it on this slide for questions, and I can move around as needed. So, thank you.
And I'll just add, we're talking to someone we're working with at Brockford on Wednesday, so we're going to hopefully develop our plan to be able to go back in and start using the lab there. And we do have the capability now to measure microsystems. So if there are water samples or things that people are curious about, we do have that capability, and we're going to work hopefully towards getting our lab certified with the New York State Department of Health. It looks like probably it'll take a little while to get through all those hoops, but we are running the ELISA method, the plate method that's sort of generally accepted by them.
So it's Sally. I know that's going to take a little while, but can you define a little while to get state certification?
Well, there's several steps that you have to go through. So I looked, and a couple of years ago, the most up-to-date stuff said that they were waiving doing some of the steps. So what we'll have to do is run several standards and make sure that we're sort of within a certain range, and then you just have to put together your standard operating procedure. So I would guess that it's going to take several months probably in order to check all their boxes and then have them come. I think they have to come do a lab inspection. And we're really at the first step, especially with the pandemic right now, because our first step is we have to come up with a plan just to be able to operate in the lab safely and not get anyone sick.
So realistically, you're probably looking at 2021.
Mm-hmm.
Yeah, to be really on the ground running with the certified. We can, as of now, we can run microsystem analysis, but yeah, for the certification, it's probably safer to think that we'll have a certified lab ready for next season.
Well, we have we know a lot of customers for you already. So let us know when you get your certification.
Okay.
Yep, we will definitely do that.
And I just wanted to let everybody know, if you have a question for Rick and Stella, you may have to unmute yourself, because I did mute everybody during the talk.
You say that there's microcystin in the foam. What does that say about the safety of being in contact with the foam compared to safety being in contact with the algae blooms?
Well, certainly, probably the best thing to do is compare them, strictly in terms of the numbers. If an algae bloom has a certain concentration and then the foam has a certain concentration, I see no reason why one would have a different toxicity than the other, because it's strictly the concentrations that we're looking at. There is, the foam, it's interesting to remember, though, that a 7-gallon bucket of foam turned into a smaller water sample. So the foam, the water in the foam is more spread out because 90% of it is air. So volume-wise, there's less, but concentration-wise, there's the same, if that makes sense. But I think that there's still a lot more to do, too, with the toxins in the foam. From everything we've looked at in the literature, foam provides just the right chemistry to concentrate things like metals and to concentrate water and soluble toxins. That's why we're doing the PCBs next. And so I think it would be good advice to say that you should always treat the foam with caution and be careful around it and not expose yourself needlessly, because there is so much more to understand.
And could you also comment on the fact that we see foam in the winter and the early spring when the water's still cold, when there isn't any algae around that we can see?
Yeah. So I think that what I think is going on, it does seem, now once again, I have no direct way to quantify this, but from the foam reports, it does seem, and from the photos, it does seem that even though foam is reported throughout the winter, in the early spring, the size of the events and the frequency of the events does not seem to be the same. And so what I would say, and once again, this is without having data from those events, without sampling a winter or an early spring event, but I would say that it is these carbohydrates lingering around, either in the DOC pool or in the mussel tissues. A lot of, when the mussels are eating this stuff and they're excreting mucus, you can often get this mucus kind of forming along the benthos, and then over time it will start to make its way up to the surface microlayer to be able to form foam. So it's probably just this kind of legacy effect, because these polysaccharides can be relatively stable in the environment compared to, and that's another reason why the algae makes them, because they are stable. They're trying to make a stable environment for themselves.
Yeah, and we have no idea if there's any toxin in foam that's forming later in the season. We don't have any samples from that, and that would be something to look at. My gut feeling is that the concentration of the toxin should decline over time, because especially if this stuff is up at the surface, it does seem that UV light helps to break the toxin down. So my guess would be that we're going to see the highest toxins when you're having active, in the foam, when you're having active harmful algal blooms. So in the late summer, in the fall. And that as the season goes on, even though you may still be forming foam, that the concentration of microcystin in it might decline. But that's really just a guess at this point.
So I guess that was something I was unclear on. I guess I understood the foam that you all felt, the foam we saw in the winter was just aged foam that had formed earlier. So you think that the foam continues to form through the winter?
That's the sense that I get, Sally, and there's a couple reasons for that. One is, from the photos I've seen, it still seems like it's this kind of wind-driven Langmuir type circulation. That's the streaking of the foam. That's what we think ultimately produces it once the chemistry of the surface is right. Okay. And so some of the photos and reports still look like that process. And most foams are not going to be stable for, I mean, really a period of days is pretty stable for foam. If anything were to stick around in that form, that would be, I mean, I don't see it happening. So okay. I do think it's continuing to be formed. Yeah.
Yeah. The foam's being formed, but the material in the surface microlayer is probably mostly generated when the algae are really active and blooming, and then that stuff's just sitting there in the surface microlayer. Yeah. Okay. So you're still there, and you get the right physical conditions with the wind and the waves to generate the foam again. And then over time, all the bubbles pop, and it moves back into the surface microlayer. You can't see it, and then you get the same physical conditions that generate it.
Okay. But I've seen, like, on not less than a month ago, a foam streak going down, sort of down towards the middle of the lake, and it, I mean, very, very visible from on a hill overlooking the lake, and it looks like it must be going for at least a couple miles down the lake. And I don't know how wide it is. It's hard to tell, but probably a good 3 or 4 feet wide at least, maybe more. And then that stuff does wash into shore. I mean, I have the foam collecting, washing into shore on my beach pretty much every couple days. So it hasn't really stopped. It hasn't stopped forming.
Right.
Yeah, and that's really, really good and important stuff to know. I mean, that's what from from what I've seen now, yeah, my best guess would be that the system is producing a lot of this stuff in the late summer, early fall, and then this stuff does stick around. And, you know, there's definitely a lot more to be learned about the direct connection between this exopolysaccharide and the microcystis and how stable it is. And is this carbohydrate breaks down? Does it need to be fresh to form foam? Or can this polysaccharide start breaking down, but it still has these kind of properties? So yeah, I think that's all stuff that hopefully we'll get more information on as we continue this type of work.
So would you like us to continue submitting foam reports on the Survey 123 form?
So yes, definitely, yeah. Yep. Yeah, that's something, I mean, since the whole system is set up, that's the kind of thing that for any research that we do after this regarding the foam in whatever lake it is in or wherever it is, that will all be very important information.
And have you thought of any other factors that might be helpful to document?
Yeah, I haven't come up with a really good way to do it yet.
Okay.
I just keep thinking of, is there any way, I mean, of course, from my perspective, you know, I want some number in a spreadsheet, like the volume of foam and, you know, reports per unit effort, like they do for fisheries. You know, it's the total amount of fish caught divided by the amount of hours that you spent fishing. So how can we do that? How can we, you know, there's more eyes on the lake during the summer. There's less eyes on the lake during the winter. So are the reports I'm getting the summer versus the winter just a function of activity on the lake? Or am I really seeing an increase in foaming events during the summer? And that's something I'm going to keep thinking about. I might try to look and see what other people have done for similar types of things to see if there's any way we can generate some real data from this. So that's something I guess we'll keep thinking about, but I don't have any discrete answers right now.
Thank
It
you.
seemed to me in the winter, because I live on the lake, there was a lot more of just streaking rather than accumulating in big clumps.
Yeah, that's really interesting, Dee. That was something I was thinking about because, you know, what translates from streaking to building up in big clumps, especially along shore, is foam stability. And so it could be that as these compounds are fresher in the summer, the foam, they're creating properties where the foam is very stable. And then in the winter, there's either less of this stuff or the stuff is broken down, or there's some other fundamental change to foam stability. So you still get the streaking, but the foam, the bubbles pop and the foam collapses quicker. So it doesn't start to build up along the shoreline in the same way. So that's all that kind of stuff is really good to know.
Or you could think, is there a temperature component to the stability of the foam? You know, does it last longer in a more, you know, average temperature, and then when it gets really cold out, be dissipated by some way? I don't know. You think about that.
It's very interesting, and it could be temperature, could be sunlight.
It could be water overturning, so just the overall stratification of the lake breaks down in the winter, and so you're really getting much deeper mixing that you just don't see in the summertime. And again, that can have to do with kind of diluting out these compounds that are allowing the foam to be generated in the first place, and so you could be kind of more thoroughly mixing the water column and mixing these compounds down, and then they'd have to make their way back to the surface microlayer, accumulate there in order to be whipped up into the foam. So yeah, that's a good point.
And when you look in the literature too, there's a lot of things that contribute to foam stability, like even like particles inside the foam that don't cause the foam but get trapped in the foam contribute to stability. So and which direction that goes in, I don't know, but the particulates change the property of the foam. So it could even be there's more stuff to pick up in the summer, and that changes its stability versus in the winter. So So these are all factors that, you know, through some data mining and through some additional testing, we might be able to start teasing out. But it certainly seems that it's related to foam stability in the summer versus the winter, and maybe due to the availability of the foaming agent.
So would it be, because I submitted photos in March and April. You know, Joel, I was on your property because you had quite a bit of buildup on your beach.
Right.
And in some areas there were the bergs, the foam bergs.
Yep.
So would there be some value in comparing a winter sample foam versus, you know, what you've collected already?
Definitely,
I think so.
definitely. Especially, I would be super interested to see what the FTIR looks like on the winter foam.
Okay.
So all of these questions are like a phase 2 for sure. You know, it's like, this is very exciting. It seems like we have more questions than answers right now, but we got a bunch of answers. So that was great.
Right.
Can you tell us? I'm sorry. No, go ahead.
Oh yeah, I totally agree. Yeah, I mean, there's my mind keeps thinking about the next thing and all these different things. Yeah, and there's many aspects of it. You know, it's so new that we're just starting to scratch the surface here.
You said something about the foam either concentrating or keeping the pollutants, some of them, or toxins, some of them we don't even know what they are. Does the polysaccharides do that themselves when they're not in foam? I mean, it was just having the polysaccharides there. Does that concentrate toxins?
Now, that is a really interesting question. It would depend on the form the polysaccharides are in. Oftentimes, when you purify something, it's going to have very different characteristics than if it's dissolved in a solution. So I don't know directly. But what I think is not in the same way. And the way I think about that is what's really concentrating the toxins in, what's really going on is that, of course, the foam is derived from the surface microlayer, the very surface most part of the water. And that very surface most part of the water has a fundamentally different chemistry from the rest of the water. So when compounds that are in the water being produced and released by biological tissues get in the water and they're not very soluble, they end up finding that surface microlayer where they are very soluble, and that's why stuff starts to collect there. So it's not the polysaccharide necessarily is like there's a direct bond between the polysaccharide and the toxins or between the metals, and that may depend on what toxin we're looking at, but more broadly, it's that surface microlayer in the foam provide an exact alternative chemistry for things that are not as soluble in water. And so that's how these things start to concentrate in there. So it's more about the solution as a whole and its properties, particularly with respect to water solubility and polarity.
Thank you.
Does anyone have any remaining questions for Rick and Stella?
It looks like Rob is trying to say something, but he's muted.
Rob, do you realize you're on mute?
Rob, you're on mute, okay? There you go.
I unmuted. There's a little red button for unmuting too, and it didn't work, I guess. Okay, you mentioned literature a number of times. We can't be the only lake who has a lot of foam forming and a lot of people concerned about it. Do you know of any, I don't know how deeply you dug into the literature, but other lakes nearby that have done research at the depth that you and Stella have done, and what their results or their conclusions are?
In this region, I've read a few reports that there have been foam sightings on Skinny Atlas and Quagga Lakes. As far as I know, there was kind of like a PSA type thing about, oh, don't worry about it, it's naturally occurring. And there certainly are, like the DEC put out, you know, a statement about that regarding the foam, but I don't know of anything on the Finger Lakes, any other research on the foam that's out there that's published in the literature. Just the few things you all have done in reading those reports. I will say they have looked extensively at foam on the lakes in Minnesota. And so some of the literature that I've read about foam and about pollutants is on the lakes in Minnesota, and they did find really elevated, sometimes 1,000 times or more elevated levels of like heavy metals in the foams that are forming out there. And I know they also have foaming on Lake Michigan, and that's related to those PFAS compounds. Again, they at first thought, oh, the foam's just naturally forming, don't worry about it. And then someone actually did a chemical study of what it was made of, and they put up hazard signs that said, don't go touch this foam. So the PFASs are just legacy compounds from industry, basically, that are just out there and everywhere now. And so probably along Lake Michigan where they're having those problems, there was some industrial dump site or something that's now leaked into the edge of the water there, and that's what those foams are generated from.
Is there some sort of correlation between quagga mussel populations and foam issues in the lake?
I have no idea.
Yeah,
and that'd be really interesting to look at.
Yeah, that's the kind of stuff that, yeah, we would be really interested to look at, and we just don't have the data. We haven't been able to find data of quagga mussel populations over time. We've seen a couple of individual surveys, but of course with the foam, you know, we're just in the beginning stages of surveying and not to the point where we figured out how to normalize the sighting data yet. So,
and there is something special about Canandaigua Lake, or maybe some of the other lakes in the region too, that allows it to have these foaming type events. I mean, Canisius Lake has harmful algal blooms, huge ones, and they don't, I haven't read reports of them getting these huge foaming events. Of course, it's a lot shallower. It is eutrophic, so it could have to do with water clarity. There's a lot of different physical variables that also have to be controlling this foam formation, in addition to just the chemistry of the foam, which is really what we focused on here. So I think that's a lot to be done, and that's why it would be interesting to look at some of these other Finger Lakes, because we really have this stretch of very similar bedrock, very similar, you know, conditions here, but provides a natural way to explore what specifically is special about Canandaigua that allows this foam to form, provided that we don't find that it is some, you know, man-made pollutant that happens to contaminate the water. And
any residual petroleum in the foam, like from gasoline and motors?
I'm sure there is, but we didn't check for that. That's why I said when we were out with Marty, and I was concerned about his boat exhaust when we were sampling, but we didn't look at hydrocarbons. So
yeah, we did when we did the so the hydrocarbons, which would be the alkanes, which is a type of lipid. So when we do the fatty acids, we start off by extracting all lipids and getting a weight, and that turned out to be a very small number. Now, we didn't look for those boat exhaust type alkanes specifically, but due to the weight of total lipids being so low, it's certainly not a sizable component.
Yeah,
it doesn't be affecting any of our data. So we might be able to go in there with good detection limits and pick it up. I'm sure we'd be able to, but it doesn't seem to be playing a huge role in chemistry.
Thank you.
Hey, Rick and Stella, I've got a question. It's Kevin Olvani here. Great job, number one. I jumped in a little bit late. You've really taken this research to a whole nother level from what Greg Boyer did and Hassett did over the years. If you go back to Greg Boyer's research, when we went out sampling, it was in 2002. That was in the June timeframe. I got to go back and look exactly, but that was when we sampled the foam during that time. It was in the June time. So when their research report finally came out, they were pointing more towards a plant-based, algae-based source as the main source of the foam production. So it aligns with what you're saying. And definitely, each time we have seen foam, it's typically that, you know, when it really explodes, it's that week to 10 days after we start to really see the first blue-green algae issues. So is it that foam? Is it the blue-green algae that's dying off that first round that begins to release the polysaccharides, and you start to see this explosion of foam that correlates with that? I guess, you know, it sounds like that's what you're pointing towards. And it sounds like it's pretty stable also, the polysaccharides, where it hangs out, because we've got aerial photos when they fly pictometry over the lake for our aerial imagery, April or November, and we have 5-6 mile length foam events in 2006, 2009, 2014, just on a random day. So we definitely see it year round, and that would be really interesting to look at, you know, the sourcing of that foam throughout the year. So that would be good to know.
Yeah, definitely. Thanks, Kevin. And we've seen, I know that we were included in the email of a couple of those photos, one from last year, and I think it actually coincided with the day that we took that sample with Marty Lasher out on his boat. We got the photo of it later that day, and it's incredible to see those long street. I mean, it's unbelievable.
I think that was a photo I took on that one, but we have like flights for our aerial imagery for the county, and I can send it just like a random day in April of 2006 when they, you know, did the aerial imagery for that year, and I can show you those photos that we've had these kind of multi-mile foaming events with the line circulation and times where we really don't have any concentrations of algae specifically. And we dominate. We dominate with Microsystem. We rarely see anything else in any type of quantity from besides Microsystem. So it's, it sounds like in the eutrophic lakes, talking with Bruce Gilman and others, that it's a much more biodiverse blue-green algae community, if you will, and Canisius and Honeoye and some of the other eutrophic lakes. And that might be, do you see polysaccharides in those other types of blue-green algae like you do with Microsystem, or is that something unique to Microsystem?
Yeah, I don't really know the answer to that. I do know that that was my thought too, is that if you have a more diverse or more eutrophic system, you also may have a different bacterial community there that can, or specialize, actually utilize these types of polysaccharides, these things that, stuff like mussels and other things, they don't have the right energy source for them, but there might also be a bacterial component that's allowing in these eutrophic systems for those materials to be broken down in a different way that's just not happening in Canandaigua because the water is so much cleaner. So that was my thought about it. I don't know about the production. I mean, exopolymeric substances are produced by all kinds of you know, photosynthesizers. Diatoms in the ocean, for example, you know, they're really important for diatoms in the ocean. So my guess is that they are being produced by those other cyanobacteria or those other critters, but that there's a more diverse bacterial community that's learned how to deal with that. And that could be something that would be really interesting to test.
Another just follow-up question with the toxin aspect in the foam. The foam, which is, it sounds like it's the carbohydrates and polysaccharides within that carbohydrate community. We've always kind of described it to the public that the foam in and of itself is harmless, but things collect in it. And that's part of the problem, is that it tends to concentrate and bring a lot of other stuff with it. We've seen on many occasions, like you were showing, clean water on either side, but we've sampled right underneath of kind of a streak of foam and found toxin levels, parallel type approach you guys did, where it was just higher than the ambient levels of toxicity in the lake water itself. Is that what's going on? Is the foam is because things can just collect and connect to it and adhere to it? That's the toxins are collecting in that foam?
Yeah, that's a great question. In thinking about this, you know, I know that proteins are pretty soluble in the surface microlayer, which is why, you know, I know for a lot of times when you talk to people, they, you know, oh, could be proteins, you know, and in some aquatic systems, you do get proteins that build up and create these foams. That wasn't what we saw here, but because the microcystin has a similar amino acid-like structure, I don't know specifically about its solubility in the surface microlayer. I haven't seen those studies, but it seems like a likely candidate for preferring that solution chemistry.
Okay.
And then, of course, it's this whole kind of thing, what's the availability of the microcystis? Is it moving along with this carbohydrate, and is it being incorporated into the surface microlayer and forming the foam with it? There are all these kinds of questions, or is it just being incorporated as algal cells and stuff washes ashore? I almost think that the way to answer that would be to, do some time series out on the lake where you kind of maybe follow a bloom or follow foam being formed and get high resolution measurements. So that's a great question, and I can theorize about it, but I can't answer it right now. But I keep thinking back to that mid-lake sample that we collected. We were out there as the foam was being formed, and it was just incredible to see that foam had, 38 micrograms per liter, even though the filtered water was undetectable. And that makes me think that if it's there, it's getting incorporated into it as the foam forms. And then you probably get, and then that additional data of the unfiltered being higher, that's probably additions to it as it's moving.